Eggers, most of you know me, I'm in the Environmental Health and Microbiology Department. We're really pleased to have two of our collaborators here this week, Dr. Paul Bradley and Kelly Smalling from the USGS, to talk about their work. And Dr. Bradley is a research hydrologist and ecologist, obviously with USGS, but he and Kelly Smalling co-lead the USGS Environmental Health Program, Drinking Water and Wastewater Infrastructure Integrated Science Team. And he's given me like a 3-sentence intro in his usual characteristic, understated way. His research focuses on human exposures to and potential effects from inorganic, organic, and microbial contaminant mixtures in drinking water at the point of use, and also on how those anthropogenic contaminant mixtures are ecosystem stressors. I'll translate that a little bit. You know, for years, EPA has done cumulative risk assessments of air toxics. Like, look at all the things that are in the air we breathe and calculate the risk together. The national primary drinking water regulations are one contaminant at a time. You know, your public drinking water supply is safe if uranium doesn't exceed its limit and arsenic doesn't exceed the limit and nitrate doesn't, and, you know, a small suite of other things. But they can each be at 90% of the limit, and your drinking water is still okay for public consumption. So for water, the EPA doesn't do a cumulative risk assessment and combine those risks. And on top of that, the standards that EPA uses for what's safe, especially for things like uranium and arsenic and lead, they're compromised between health standards, what we really know about the health risks, and what's technically and economically feasible. So therefore, the standards are, you know, for arsenic, it's 10 micrograms per liter, when really we know no amount of arsenic is a safe exposure. About the same for uranium and for lead. So what this team has been doing and publishing since about 2018 is a number of different studies around the country to look at point of use water consumption, both from home wells and from public water supplies, and really including home wells, I think, for the first time in this kind of risk assessment. And not only doing a cumulative calculation of what are those combined health risks, and then secondly, also using health standards of, using really low values for arsenic and uranium and lead, getting as close to zero as possible, instead of using the standards that are based also on economics. And that's showing pretty high risk, substantial health risks for drinking water supplies across the country, both in public and in home wells. And they've also recently done a study on bottled water. So Dr. Bradley is here to talk to us about this research, and I just have to say they're a pleasure to work with, and we're really enjoying working with them on our working pro, and it's just a delight to have you guys here. So that.
So that pretty much covered it, so we can have questions. So I don't need to push anything.
Nope.
Okay. All right, so real quick here, USGS, 5 mission areas, 22 programs. One of the mission areas is ecosystems. One of the programs is environmental health. This research is environmental health. So Kelly Smalling over here and I are the national team leads for a project in environmental health programs, which is the drinking water and wastewater infrastructure project. So it's basically the human impacted water cycle. So for convenience, we break it down into 2 focus areas. One is the impacts of anthropogenic chemicals in stormwater and wastewater, those impacts in aquatic receptors and associated terrestrial environments. And then the other side that we're focusing on today is the focus on the human as a receptor, primarily exposures through ingestion of drinking water. So it really doesn't matter whether you're focused on the environmental side or you're focused on drinking water side. One of the fundamental problems that you're facing is the same. Whatever your metric is, whether you're focused on the TSCA inventory, 85,000 plus contaminants, the Wang et al. Publication is estimating globally 350,000 in commercial production. So that doesn't include any environmental transformation products or degradation products, right? That's just the parent compound. Or the cash registry, that number is really huge compared to that number. So depending on how you count them up, national primary drinking water standards address circa 90 contaminants, right? So that's the fundamental issue. That's a theoretical problem. In the real world, the evidence suggests that that is in fact the case. So this is a study that was conducted a decade ago by the USGS and the EPA. The focus of this study was to kind of move contaminant effects science away from this single contaminant or single contaminant class point of view towards the reality matrix, which is contaminant mixtures. So the effect on you is everything you're exposed to. So in this study, the focus of the study was to combine all the analytical capabilities of both agencies. And amongst other things, we analyzed more than 700 unique organic compounds. More than half of those compounds were detected at least once. And of the ones that are detected, more than half of them are actually compounds that are designed to be biologically active. So pesticides and pharmaceuticals being the largest group of those. So the plot here is we got 38 sites oriented along the x-axis. These are surface water sites across the nation. On the y-axis, unlabeled, are half of the detected compounds, and each bar represents a detected concentration. And we know that this kind of contaminant complexity is making its way through existing drinking water technology. So this is another USGS EPA collaboration looking at 25 drinking water treatment plants across the nation and comparing raw intake water to the finished drinking water. They assessed 247 compounds. This is organic and inorganic constituents, 148 in the intake, 121 detected at least once in the outtake. So what's being distributed. So one of the fundamental problems, there's obviously a lot of focus on what's the role that drinking water contaminants play in human health. And so in 2016, we had a national meeting with other federal agencies and state agencies and university researchers who are focused on the public health side and trying to understand the role of drinking water contaminants and ask them, what's your fundamental problem? Well, the fundamental challenge is they're trying to estimate human exposure through drinking water based on water quality data from USGS, for example, and the EPA, which is collected in an ambient reservoir. So it's collected in the surface water environment or it's collected in the groundwater environment. And that is miles and years separated from the actual exposure point. So what exactly are you being exposed to at the point of exposure is the primary limitation for this kind of research. All right, so it's there at the intake, it's coming through the outtake. Our research is focused specifically on supporting public health researchers, decision makers, and most importantly, the communities that are being affected by examining exposures with two criteria. One is we're doing it at the point of exposure, so we actually go into people's homes to collect the samples. And we're using an expanded analytical toolbox based on this national recon that we did back in 2012 to 14 that more realistically reflects what we see in the environment. So a quick background here, 3 distribution pipelines for drinking water in this country: public supply, private supply, commercial or bottled water. Commercial bottled water and public supply are regulated. There are differences in how they're regulated. Public supply is regulated as drinking water. Bottled water is regulated as a food. Okay? Both of those regulations focus on the regulated contaminants, and most of their assessments are done pre-distribution. And then for private wells, obviously EPA is not authorized to monitor private wells. And so the cost of monitoring maintenance falls on the homeowner. And as a consequence, we really have very little information on exposures in private well water. So the approach we use has a core analytical toolbox shown on this slide. Each sample consists of collecting about 30 bottles. We can do it in about 15 to 20 minutes because we've had lots of practice. And then we analyze each of those samples for roughly 500 unique organics. These include cyanotoxins. These would be associated with harmful algal blooms, disinfection byproducts, mostly associated with public drinking water supply. Occasionally we see them pop up in private wells. Pesticides and pharmaceuticals we talked about earlier. They're designed to be biologically active. PFAS is on everybody's radar these days. And then the volatile organic chemicals. This is the chemical class that most of the historically regulated drinking water contaminants fall. And then we also look at the inorganic side. So we do major ions. Concerns would be things like nitrate, nitrogen, and then trace elements. And we like to combine all our studies with microbial indicators as well, because there's generally a trade-off between some of these compounds and exposure to microorganisms. So let's say the analytical space here is quite large. So we're analyzing for a lot of constituents. The cost per sample is quite high. We're limited in the number of samples that we can collect and analyze in a given year. So we tend to conduct our research on a community basis, so community by community, in order to compare those results across communities and across years to develop this kind of aggregated national perspective. We try to control for variation from study to study, so we use that same core analytical toolbox, and we have a team that goes into the field to collect the samples, and then we use the usual USGS quality control quality assurance. So a quick context. We are not assessing the drinking water quality, the source water quality. We're not assessing the quality that's coming out of a drinking water treatment plant, right? What we're trying to assess is exposure to humans. So when they go and they turn on the tap and fill up a glass with water and drink it, what ends up in that glass? That's what we're after. So contrary to compliance monitoring, we're not going to police the area. We're not going to take that screen aerator off. So if you haven't cleaned that thing in 2 years, that green biofilm is going to end up in that sample, right? We're not going to clean the sink area, and we are not requiring the stagnant overnight, 6-hour stagnant overnight protocol for lead and copper. So you can't interpret our results as being the worst case scenario for lead or copper. All right, so we've been doing a lot of studies since 2016. We'll go over 3 examples here today. We'll start with Cape Cod. So Cape Cod this study was specifically designed to kind of balance our understanding of public supply exposures versus private supply exposure. So at Cape Cod, the source water for public and private supply is the same shallow, sole source, unconfined aquifer. It's a sand aquifer system. And it has documented septic impacts on the island, and it has PFAS impacts. Because someone asked me, right in here is Joint Base Cape Cod. All right, so the study design was 10 public supply locations spread across the island. They're shown here as the squares, and 10 private well locations. These are the results. So of the 487 unique organic analytes that we analyzed for, 70 were detected. They range from 1 to 17 compounds in public supply, 2 to 30 compounds in private supply. Fairly similar detections, fairly similar range of cumulative concentrations. There were some notable differences. So if you can't see it real well, public supply is on the left-hand side, private supply is on the right. This collection of 21 pharmaceutical contaminants was in a single tap water sample in a private well. So the problem here is they're down gradients of somewhat septic tank. This is the same results. They're broken out here by individual classes. For each of these, the contaminants are ranged on the x-axis, concentration or total detections in the upper plot on the y-axis. The shaded box is public supply. The open boxes are private supply. And there are a few notable differences here. One is 6 of the public supply locations on Cape Cod actually treat by chlorination, so chlorine disinfection. So for those 6 locations, disinfection byproducts are by far the biggest contributor to the detected signal. Then this is the same example before. You've got this private well outlier that has 21 pharmaceuticals in it. And then we have PFAS contaminants. At the time this study was done, Massachusetts had an MCL for the sum of 6 PFAS compounds equal to 20 nanograms per liter. There was 1 exceedance in a public supply sample, and 3 private supply samples exceeded that standard. Now, obviously, last week, EPA dropped proposed MCLs and MCLGs. So if you go back and kind of retrospectively characterize those results, you'd have 5 locations that exceed the PFOA standard, proposed standard of 4 nanograms per liter. And then 4 locations would exceed it for PFOA. The important thing is the MCLG that's proposed for PFAS is 0. And so half of the samples on Cape Cod had detectable PFAS concentration. So we're trying to understand exposure, right? So half of the risk element is the exposure, and then the other half of it is contextualizing it by the hazard. So the way we're contextualizing the hazard is we're calculating, in this case, exposure activity ratios, and those are the measured concentration divided by the activity concentration at cutoff. This is based on data in ToxCast, okay? And so I think it's on the next slide. Let me cheat to that real quick. So the advantage to ToxCast is that it has more than 10,000 organic chemicals have been assessed at 1,000 approximately molecular endpoints. And these endpoints are specifically designed to inform human health, so they're vertebrate-centric molecular endpoints. So the fact that we're looking at molecular effects level is potentially more sensitive than looking at an apical effect, but then there's that question about whether or not the molecular effects are actually going to transfer up to the apical level. And then, as we can do for any of these kind of toxicity-weighted risk assessment methods, you can calculate the individual exposure activity ratio, and then you can add up those individual years to get a cumulative estimate of risk. So the plot here, and we'll see very similar plots to this from here on out. Public supplies on the left, private on the right. The sites are not labeled, and they're along the x-axis. Exposure activity ratios and log space are on the y-axis. Each dot represents an ear for an individual compound, and then the red triangle at the top represents the cumulative ear for that sample. So for ear, we use 2 different kind of screening levels. The exposure activity ratio equal to 1. For a single contaminant exposure activity ratio, this means the concentration is equal to the in vitro effects level. That's a really simple, straightforward interpretation. We also use a precautionary screening level of exposure activity ratio equal to 0.001. The reason we use something that's 3 orders of magnitude lower is because the ACC, unlike human health benchmarks, does not have any built-in margin of safety in it. So typically margin of safety for human exposure involve 2 to 3 orders of magnitude. So that's one reason for it. Another reason for it is we know we're underestimating exposure, because for this study, 487 unique organic compounds were assessed, which seems really large, and it is, by most studies that are published today, but it's really small compared to whatever your metric is of the total number of contaminants being released. And then for this study, roughly half of the compounds that we detected actually had data in ToxCast. So there's this built-in underestimation going on. So we use a precautionary screening level. We use 0.001. In this study, you can see that 2 sites, both public supply, actually equaled the in vitro effects level, so that's a concern. And then all sites but 2 public supply exceeded this precautionary screening level of 0.001. So the fundamental challenge to the ToxCast approach, again, molecular level, potentially variance sensitive, and it's telling us about things down at the starting point, potentially for human effects. One of the fundamental limitations to it is only organic contaminants. It doesn't contain inorganic. So at Cape Cod, we have concerns about inorganic contaminants as well. So they're ranged along the x-axis, again, concentration on the y-axis in a log scale, looking at the left axis. And the public supply is shaded. So we have, for example, copper and lead. And so the National Primary Drinking Water Standards, or the action level in the case of lead, are shown as dashed lines. And so you can see that there are private well locations that are actually exceeding those standards for lead and for copper and for nitrate. There were no exceedances on Cape Cod of any MCL. But then the MCLG for lead, as an example, is 0, and half of the sites at Cape Cod had lead exposure. So inorganic exposure is also a concern for risk. So we use an analogous approach to the exposure activity ratio. This is the kind of classic toxicity quotient or hazard quotient approach. And we're, in this case, you're dividing the measured concentration by an apical human health benchmark. This benchmark has a margin of safety built into it. So the interpretive thresholds are going to be different. So the risk screening level for TQ is TQ greater than or equal to 1 is generally considered a high level of risk to human health. And then the cutoff for low to no risk expected is 0.1. So every sample at Cape Cod exceeds the cutoff for no risk, and all but 3 sites are exceeding the high risk threshold. So this study has been published. My apologies for not pointing it out earlier. Hopefully you're all tech savvy. So these QR codes will actually take you to the publication. So you can just scan them with your phone and it'll create a web page for you. So the next example is next door. Collaboration with Northern Plains nations in North and South Dakota, conducted in 2019. So the Strong Heart Health Study is a cohort study that's been taking place since the 1980s, specifically focused on understanding environmental contributors to cardiovascular health in the American Indian populations in Arizona, Oklahoma, North Dakota, and South Dakota. One of the early findings in those studies was the connection between adverse health outcomes and arsenic exposure in drinking water. And as a consequence, the communities in these states have been trying to do interventions to reduce exposure to arsenic through drinking water. So in North Dakota and South Dakota, for example, 2 interventions are taking place. One is connecting people on private wells up to community water supplies, which are then regulated and monitored. The other approach is an offshoot of the Strong Heart study, which is the Strong Heart Water Study, specifically designed to go to rural homes that can't hook up to the public supply and install point-of-use treatment to reduce arsenic exposure. So we connected up with the researchers on this project. We explained what we're doing in kind of this broad contaminant mixture exposure paradigm, and came to an agreement to collaborate on this study specifically to support their ongoing activities and to support the community's decision-making by giving a broader perspective of what other contaminants you might be exposed to other than just arsenic. And that would give you the opportunity to simultaneously manage for things other than just arsenic and potentially make upgrades and adjustments to your treatment paradigm. So this initial effort in 2019 was just a reconnaissance level effort, and it was a convenient sampling of locations that were already built into the Strong Heart Water Study. All right, so in 2019, we collected a total of 56 tap water samples. 34 of them were from private well locations. Those are shown in the circles, and 22 are public supply locations. So we started out in Community B. We're anonymizing these results. So Community B, and then we went to North Dakota in October, and then followed it up, racing the weather and winter for the last community in South Dakota. So these are the results. We'll go through these one by one. But to orient you to the slides, communities A, B, and C, so that's from the southwest corner up to the northeast, are along the x-axis. Concentrations are in log space. On the y-axis for individual contaminants of concern, in this case inorganics. When public supply samples were available, public supply locations were only sampled in communities B and C. Community A, we only have permission to collect samples from private well locations. And those private well locations were specifically part of the Strong Heart Water Study, which means their entrance criteria for participating in that study was an arsenic concentration of 10 or higher. So that's kind of a built-in bias in Community A. So public supply is shown in blue for communities B and C. The white boxes are all private well locations. And on these plots, if there is a EPA enforceable standard for public supply, it's shown as a red bar. Obviously, it would not be an MCL for lead. For lead, it would be an action level. So the short message for this study, this collaboration, was 30 inorganic constituents were detected. There were no exceedances of any MCL for public supply. There were exceedances of the MCL equivalent concentration in several private well locations. So this points out the inherent risk of private wells. You don't really know what you're drinking. And then we had several cases where both public supply and private supply are exceeding health advisories. So walk through these because this is next door and potentially has relevance to Montana. Arsenic concentrations. The MCL is 10, as Mari said. 14 of the 56, or 25% of the samples in this study, exceeded the MCL. They were all private well locations. So these are individuals who are not aware of what they're drinking. In terms of the private well samples, that's 42% of the total private well samples collected. So there's concern that 10 micrograms per liter is not protective enough. There are a couple of states that have already instituted MCLs that are lower than that. New Jersey and New Hampshire are set at 5. Every site in this study that had a detection of arsenic exceeds that standard. And then the health advisory obviously is 0. And so again, more than half of the samples in this study are have detectable concentrations, and so they're exceeding that MCOG. All right, uranium. Uranium's MCL for public supply is 30. One sample in this study, a private well location in Community C, actually exceeded that. The health advisory is 0, so about 43% of the samples in the study had detectable uranium. So uranium, in addition to arsenic exposure, is a concern in these communities. Lead. Again, keep in mind, we did not follow lead and copper rule stagnant sampling protocols. You can't assume these results are worst case scenario. So no exceedances of the action limit for public water supply, but we do have the MCLG of 0, and 20% of the samples had detectable lead. So lead typically is showing up in drinking water as a contaminant coming from the infrastructure itself. Right? So it's either coming from, generally from the distribution pipeline and public supply, or from the premise plumbing. And the private well locations, it would be the plumbing from the well structure up and into the home. A common source of it is copper pipe, copper plumbing using lead solder. And or you're using old fixtures at your faucet which contain high brass content with high lead content to the brass. Nitrate, nitrogen, the MCL and the MCLG is 10 milligrams per liter, 3 orders of magnitude up from previous slides. One sample, again, private well, exceeded the MCL. The MCLG is the same. The kind of context to this is that the human health impact that's driving the MCL and the MCLG is methemoglobinemia in infants. There is a lot of more recent research which is focused on adverse health effects, including cancers at lower concentrations than the MCL. So those concerns exist down to 1 to 2 milligrams per liter. Manganese. There is no MCL for manganese. EPA does, however, have a health advisory. So this is a lifetime exposure. The way this works is this is assuming 100% of your exposure comes from drinking water. In this case, manganese is focused on adverse effects, neurological effects to children and infants. And that value is 300 micrograms per liter. And that value was exceeded in 32%. So about a third of the samples in this study exceeded that value, including the majority of samples in public supply and community C. The WHO, World Health Organization, has also distributed a provisional guidance value of 80 micrograms per liter. Again, the concern here is of neurological effects on infants, specifically bottle-fed infants is the concern that's driving this one. And 39% of the samples across this study exceeded that level. We get to, and yeah, the final closing comment here is there's also an acute standard for exposure of small children. So these are children less than 22 pounds, less than 10 kilograms. And the 1-day acute exposure level for those is 1 milligram per liter. So you actually have a sample here collected that exceeds that value. This also happens to be the same sample location where the uranium exceeded the MCL of 30. So you've got uranium and manganese concerns, especially for small children. Selenium. MCL is set at 50. The MCLG is the same. And there were no exceedances of that value. There's been some research questioning whether or not that needs to be lower to be more protective. The European Commission actually put together a provisional value of 20 micrograms per liter. That would have been exceeded by one sample in this study. Interestingly, there's a kind of a cohort study of opportunity taking place in Italy, where there was a community that was exposed to higher than surrounding area, higher selenium concentrations than the surrounding public supply drinking water. So these are all public supply. The elevated selenium exposure that this community was exposed to, the researchers documented adverse health effects, neurological effects, neural tube effects, a number of other effects related to that high exposure. And what's interesting about that is it's at the high exposure they're talking about is 8 to 9 micrograms per liter. That same research group suggested the safe drinking water level for selenium is 1 microgram per liter. So 16% of the samples in the study exceed that. More than half of the samples from community A are exceeding that value. So selenium is potentially another unrecognized health concern driving these communities. Organic results. The circles again are private wells. The diamonds are your public supply locations. The color coding is darker code is higher cumulative concentration of organics detected in the sample. The size of the symbol indicates the relative number of detections. 476 organics were analyzed in this study, 63 were detected. There were no exceedances of MCLs. That's not as impressive as it sounds because most of these don't have regulations. Health advisory levels, there are some compounds that have MCLGs of 0, so a detection is a nominal exceedance. And public supply locations, as we saw with the Cape Cod study, this is primarily disinfection byproducts, 3 of which have MCLGs of 0. And then the others are single detections of volatile organic chemicals in private well locations for these compounds. So we're taking those organic results, and as we did for inorganics, we're displaying them by class. Again, communities on your x-axis, concentration still in log space on your y-axis. And in the upper right, you have the cumulative concentration. Each symbol in the upper right represents the cumulative concentration of detected organics for each sample. Move over to the one on the upper right. The class is now disinfection byproducts, and you can see for public supply locations in community B and C, disinfection byproducts are basically what you're seeing, right, at those sites. They're responsible for more than 99% of the detected concentration. The other interesting results here is you move down to the lower left plot. You have high pesticide concentrations in 7 locations in Community B. 3 of the locations in Community B on public supply are from groundwater wells. 7 of them are sourced from the Missouri River. The Missouri River has pesticide signature in it, and so that pesticide signature is showing up at people's taps. And then the highest concentrations detected in this study were actually a volatile organic chemical, is isopropyl alcohol, detected at a private well location in Community A. So for context, for disinfection byproducts, the THM standard, the MCL is 80, so public supply is below that. So again, going to contextualize these results, exposure is only half of the risk paradigm. So starting out with the ERA calculation, measured concentration divided by the ACC, the activity concentration and cutoff. Same 2 thresholds for screening. 1 sample in Community A actually exceeded an ERA of 1. I think this is the highest we've seen in any study. And then in terms of the precautionary screening level, roughly half of the samples in this study exceeded the screening level of 0.001 for concern and attention for further study. Doing a toxicity quotient approach, analogous approach specifically to bring in the inorganic constituents. When you add in the inorganic constituents, again, TQ greater than or equal to 1, high risk of human health effects. All but 4 samples are in that zone. So all but 4 samples have a high risk of human health if you include inorganic exposures. And then the cutoff for no risk is 0.1, and every sample exceeded that. Again, this has been published. You can get to the publication here. If you have any trouble getting these publications, I think they're all open access, but if you have any trouble, just reach out to us. And then, so we focused so far on public supply and private supply, and we find it increasingly uncomfortable not to balance out the story. So we did a study in 2020. Coincidentally, we're under COVID restrictions, so we can't go anywhere. Certainly can't go into people's homes, right? So we purchased 30 different bottled waters. 23 of them were domestic. These are all available in the U.S. through online purchase, which is the constraints we were operating on. So 23 of these bottled waters are from the U.S. 7 of them were imported. Of the 23 that were from the U.S., 7 of them are actually purified tap water. So this is just tap water that's gone through an extra filtration step. In this case, all 7 of them were subjected to reverse osmosis. Okay? The imported samples are going to be shown in blue color, and then the white is going to be the domestic samples. It's worth pointing out, so if you're a fan of FDA labeling, FDA has lots of categories of bottled water. Purified tap water is one, and then there's artesian and spring water, etc. Everything that's not purified tap water is under the label spring here. Again, the blue is your imported, the white is domestic imports and domestic for spring water samples only. We're looking at just 4 examples of inorganic exposures. First thing that comes to mind is purifying your tap water is better than bottled water. So perhaps as attractive as spring is, that's not what you should be gravitating to if you're going to buy bottled water. The other things that are important to keep in mind is, again, no exceedance of, in this case, it's FDA, so it's not an MCL. It's a standard of quality or shall not exceed level. For the most part, and the federal regulations require that when EPA promulgates an MCL, FDA has a very short window of time to make a decision on that. And they either promulgate an MCL or explain why it's not relevant to bottled water. For the most part, they promulgate the MCL as a SOQ for FDA. Okay. There are exceptions. Lead's an exception. The MCL or the SOQ for lead in drinking water is 5. Can't exceed 5. So what this is showing you is in bottled water, you got nitrate exposures. You got nitrate exposure in one bottle of water, which is just sitting right below the MCL. Again, we said there are health effect concerns going down to about 1 to 2 milligrams per liter. So you have bottled waters that are exceeding that. You have arsenic exposures above 1 microgram per liter, in one case just below the MCL. So bottled water. Uranium, same thing. You got several bottled waters that have detectable uranium, and 5 to 6 of these actually were above 1 microgram per liter. And then for lead was detected in both purified tap water and spring water bottled waters. This is a really low number. There shouldn't be any lead, period, in a bottled water facility. Organics detected in the study: purified tap water on the left, spring water on the right. Projecting this into effect space, starting with the EAR values. You actually had 2 spring waters that exceeded an EAR value of 1, so this is exceeding the in vitro effects level. And then you have several, about half of the bottled waters are exceeding this precautionary level that we're using for level of concern. Drop over into TQ space, looking at apical effects in humans. And all but 2 bottled waters, 1 spring, 2 spring and 1 public supply, are exceeding the TQ equals 1, so a high risk of exposure. And then only 1 sample actually fell below the threshold, 0.1 threshold for no expected risk. So the message is beginning to unfold here. This is a shared challenge across all 3 distribution pipelines. There is no safe refuge. It requires you to be engaged in your drinking water supply and pay attention. To drive that point home, here are 3 examples. So we got nitrate on the top, arsenic in the middle, and VOCs on the bottom. Private supply, this is the samples collected across all of our studies. So private well locations, public bottled water locations, you look at nitrate, very comparable between public supply and bottled water. They don't exceed MCLs. That's what they're focused on. But the exposures are still there. And then private well locations, similar median concentration, but you still get these exceedances because this is unmonitored water, so you get exceedances of that MCL. Arsenic, similar distribution for public supply and bottled water. Again, they're paying very close attention to those standards. And you're getting the exceedances in private supply. So a real breakdown in arsenic in private supply locations, depending on whether arsenic's part of their geochemical environment. Lots of non-detects, but then you got lots of locations in North Dakota, for example, that are exceeding the MCL. And then VOCs, same general message, very similar distributions between bottled water and public supply and private supply as well. With private supply, you get these outliers because it's unrecognized, unmonitored. So the take-home message here is human impacted water cycle. We are impacting our drinking water. It exists across all 3 drinking water distribution pipelines. And the solution to the problem requires us to focus on the source of contamination and regulation across all pipelines, as opposed to assuming that one of these distribution pipelines is actually the answer. This again was just published, I think, last month. You can get to it through the QR code. So we've done a lot of these studies over the years. The ones that are shown here that are in black font have been published out, should be open access and available. The Minnesota and California, or the Minnesota and the National PFAS Recon that Kelly presented on yesterday are in submission now, should be coming out shortly. The rest of these are in progress studies. If you have any questions, you can reach out to the 3 of us. And then this last site is a geo-narrative or a story map, if you prefer, where you can go and get kind of one-stop shopping. To date, it has maybe 5 of the studies. We're going to load 3 or so more studies pretty shortly. So the idea behind this is you can go to this one location, you can follow all our studies, and you can also get a national perspective building out of these individual community-based modules. All right, I think that's it. Happy to answer any questions.
Thank you to Dr. Bradley and team for a great talk. What I was interested to learn is how did you translate this to the individuals where you collected the water in their home?
Yeah, so this research is conducted specifically in cooperation with communities, but it's also a cooperative agreement with the individual participant. So when we collect a sample in their home, our promise to them is that we will return their individual results. At the community level, we return results to the community as well, but those results are all anonymized, right? So the community can see across the community what ranges and concerns are, but they don't know what anybody's result is except their own, right? So once the analyses are complete, it goes through a USGS approval process. And once it's approved, then we will create an individual homeowner letter and send that report to them. We like to do most of these community research efforts in cooperation with a community liaison. It can vary from study to study. In the Dakota study, it was Missouri Breaks Industries, which was a private nursing company. And so we sent the samples. They were part of the research team. They knew who each of the samples came from. We sent the individual homeowner letters to Missouri Breaks. They would drive out to the participant and give them the homeowner letter and then answer their questions, right? And each of these homeowner letters has our contact information. They can always contact us and we'll go through this stuff. And then typically we'll do several community briefings where we'll actually go over the results for the community at different levels. That can be, in the case of the Dakota study, we gave a briefing to each of the 3 different tribal councils for the 3 communities. And then also presentations to the public at large, if that's the way the council wants it to go. And then we are a science agency, and our research is funded by public funds. So we are obligated and mandated to release our results to the public. And so we publish this data in scientific journals as well as on things like this geonarrative. Again, we're going into people's homes, so this is private information, right? So all of these results are anonymized. So there'll be a de-identified geo-reference for the location, but that location information will not get you to the actual samples. Yeah.
So kind of asking you to predict the future here for two different dimensions. So first dimension is, longitudinally, if you would go back to the exact same sites, take another sample. Could you talk a little bit about that? And then could you talk about that also in the context of aquifers sort of not getting refreshed as much as they should, and maybe climate change and how that might influence these results?
Okay, so
I realize you might not have data on all that.
What's the first question?
So the first question is just repeat sampling, assuming that everything
Yeah,
is
so
steady
temporal, right?
state. Yeah.
So, and we're at the cutting edge, right? So the question when we started in 2016 is what is exposure, realistic exposure at the point of exposure? Do you have to go to the point of exposure, or can you do it at the source like they do in public supply? And so that has been answered, right? So now we're basically trying to understand what exposure is across the nation. And the sample design for that, again, the caveat here is analyzing each sample is very expensive, right? So we're limited in what we can do. So the golden grail of infinite funding doesn't exist in this equation. Right? So to answer the question of exposure broadly to the population, spatial assessment, not temporal assessment. So these are all spatial. They're synoptics, they're one-times. The temporal variability is an open question, and it's a big concern. We have done aspects of temporal sampling in a few of our studies. For example, we did a study in Puerto Rico where we went to 2 locations, one a residence and one a commercial location. They're both on public supply, and followed them over 3 days. A first flush sample, 30 minutes later sample over a 3-day period. And there were some pretty dramatic differences. For example, disinfection byproducts, 100% change over a few days. So you would come in and out of regulated space. You would pop above an MCO. Things like lead, as you might expect, lead exposure above the action level, stagnant, 30 minutes later flushing, it's gone. So yes, there is temporal variability. If you were here for Kelly's presentation last night for the PFAS recon, which is just focused on PFAS, so it didn't do all the other contaminants, we measured up to 80 days at 3 different locations. And for PFAS, there wasn't a lot of difference. There's some difference in detections of compounds, but that has more to do with our detection limit. They pop in and out of our detection limit. Right? So yeah, temporal is a big one. Eventually, we'll need to get to a point where we identify a location, set up and go longitudinal. But right now, we're trying to, if you will, right-size the perspective for the U.S. population, and by extension, the global population. And the second question was?
Well, is that aquifers don't get refreshed as often, or is to the extent?
Yeah, so climate changes, so long-term trends, and broaden that question out to just trends in general. So yeah, I mean, the climate effects, obviously, if the water table drops the zone you're drawing from is going to change, so your chemistry may change. Related to that, potentially more of immediate importance is these private well locations, these are all pumped wells, right? Which means they're drawing down. And the design criteria for a private well is water. So you go as deep as you need to get reliable water quantity. You know basically nothing about the quality, right? So to get the water quantity, and no deeper because you're paying by the foot. Right? And so, and you leave it in order to ensure the most water possible out of that well, you leave a large open hole interval, which means that your capture zone is going across potentially multiple fractures, or in an unconsolidated system, multiple redox zones, right? So each day you're pumping that well. Beginning when you turn that pump on, you were capturing all of this. As the day goes down, you're now capturing this zone. So the change in zone supply is changing daily. But perhaps most importantly, if your private well isn't very productive and you really draw that well down, you have now changed the redox zone for that entire open hole. And as it comes back up, the chemistry is going to reflect that, which is why you can get into areas like in the Dakotas or a shallow private well where you have arsenic and uranium together. In theory, you wouldn't expect it, right? Because the uranium mobilized oxidizing, arsenic reducing. But the reality is it's a mixed heterogeneous redox zone. So you're getting iron, manganese, selenium, uranium, arsenic. Yeah.
I'm sure the human health consequences are, you could talk about forever, but just to summarize at the broadest level, are there common human maladies that you're pretty confident relate to exceedances of certain chemicals? How much at risk is human health in places where the exceedances are happening?
So in public supply space, the MCLG is specifically just focused on human health. It does have a margin of safety, and it's also focused specifically on the most vulnerable populations, right? So it's going to unborn infants, pregnant women. It's literally, that's where it's going. It's trying to define the safety zone. So everything that has an MCLG defined, that process involves compelling evidence for human health effects, apical level human health effects, right? So those are health benchmarks. So it's literally something that's going to show up in a whole organism. It's not a molecular level signal that's been tweaked. Okay? So the safety is there. Basically everything that we discussed has in research space has evidence of associations with adverse health effects. Often it's multiple cancers and neurotoxic effects. So in research space, several compounds like, nitrate's an example. So nitrate's regulated for methemoglobinemia, but there's research that's more concerned with cancer effects, and that's down in that 1 to 2 range. So if you're talking in regulatory space, if it has an enforceable standard, it's definitely a concern, and it has demonstrated human health effects. Then the question is that MCL actually sufficient to cover everything? And you can make up your own mind about that, but research space says not for many of these compounds. Selenium is another example. Manganese is another example. It will take generally a decade before something with compelling research evidence makes it up to a federal regulatory standard. So there's a significant lag there. And states are typically will move quicker than that if there's a demonstrated concern, which is why, for example, New Hampshire and New Jersey are already at 5 for arsenic. So, and then it goes back to the introduction that Mari made. That's still thinking of things, contextualizing things in terms of individual contaminants relative to their safety threshold. But the reality is adding them all up, they interact somehow to give you an accumulative exposure. And that is the unknown. We don't know. And these mathematical calculations, computational toxicology calculations are designed to give us insight into it, but the question is still open. If you really want to know, you gotta reconstitute one of these mixtures and do a toxicity test, right? And then every single sample has a different mixture, so.
Should we think about wrapping up, Lori? Another question or so, and then we'll close out?
I guess I'm curious about sort of the retroactive MCL idea. Like, I think about pesticide cocktails that we know occur in our groundwater, and they're all, we might have to date thought about them individually. At what point, or, I mean, you're saying we don't know, there's a different suite in every sample, but we know that there are cocktails of 30 to 50 compounds in shallow groundwater in parts of the state right now. So how, do we wind up going back to those analyses and I don't know, thinking about a restriction level to apply retroactively? Or I guess maybe the more important thing is to be precautionary in terms of where we're headed with groundwater contamination in particular.
Yeah, and I'll couch this response. We're in research space now, right?
Yeah.
We're way out in front of regulatory space.
Sure.
And so starting back with the 2012 to 14 study with EPA's, that mixture study was specifically designed to boot us into exposome space, if you will.
Right.
Acknowledging right up front, 500 compounds is not the exposome, but at least it's more realistic. But in terms of how we regulate that, we're not there yet. There's kind of a precedent for how you might regulate, right? The disinfection byproducts have two rules. One is trihalomethanes, so they're regulated as a class. And it's been suggested that you could regulate PFAS as a class. And there are lots of challenges to making that actually happen. But to regulate the risk overall, you need to do some sort of cumulative risk assessment. You need to start asking these kinds of questions. And the answer to your question is, we're still at the research level, we're not at the implementation.
Maybe there's a middle ground though in protecting groundwater is the main thing I think about with managed recharge, where we're thinking about water supply, and yet we're also compromising the resource in ways that we don't totally understand. Perhaps there's a way to target those kinds of activities.
Yeah, that's very true. And I mean, so managed recharge, indirect potable reuse, direct potable reuse, if you're in the Southwest, the water you're drinking has probably been cycled 3-4 times. Yeah, I agree. We need to move in that direction. The easy message is protect your water. And the other easy message is, once you have done studies like this where you've done a quantitative assessment that you can directly compare across public supply, private supply, and bottled water, and you've eliminated the illusion that the answer is to go one way, then maybe people recognize it's a problem, period. Let's try to find solutions, like improved treatment, whether it's in a bottled water plant or a drinking water treatment plant.
Right.
Improved communication to private well owners so that they can think about prophylactic treatment. They can't afford to pay for analyses like this individually, but they could potentially invest in point of use treatment, and maintenance of that, given these kinds of concerns. And then along those lines, in New Jersey, kind of leading that discussion too. You can start talking about researchers in New Jersey, not the state. You can start talking about some sort of regulation for private supply, or if not regulation, at least a monitoring program for private supply, where you can get kind of regional understanding of what risks are. Because that's about 14% of the US population, about 40 million people, who are just a total shot in the dark.
Well, thanks again.